First up, we have Alterity Therapeutics Limited, ASX code ATH, and they have a market cap of around 19 million. Alterity Therapeutics is a clinical stage biotech company dedicated to creating an alternative future for living with neurodegenerative diseases. Presenting for the company today is CEO Dr. David Stamler. David, welcome to the network, and over to you.
Well, thank you, Paul, and thank you for all those attending in Australia and the US and elsewhere. I am very happy to tell you about the great progress that we've made at Alterity in 2023 and the excitement that we have for looking forward for the remainder of this year. Next slide. These are our forward-looking statements. I would encourage you to check our regulatory filings. Next slide. So as Paul mentioned, we are dedicated to finding an alternate future for people living with neurodegenerative diseases. And our mission is really in our name. Alterity actually means the state of being different. And what we're really trying to do is change the state of individuals who are living with disease to health. And we're doing that in a way with our drug candidates. Next slide. So our strategies for developing new therapeutics here are to develop drugs that modify the course of disease. Our lead drug candidate is called ATH 434. I'll refer to it sometimes as 434 during this presentation. And this is a novel drug candidate that we discovered in-house, actually in our labs in Melbourne. And this drug targets the proteins that are implicated in neurodegenerative diseases like Parkinson's disease and related disorders. Now, our first indication is a Parkinsonian disorder with no approved treatment. And that disease has a name called multiple system atrophy, something you've probably never heard of before, but it looks a lot like Parkinson's disease. I'll also call that MSA. This is an orphan disease based on the small numbers in the particular regions where it's been characterized that way. And that affords us various opportunities with the regulatory authorities. Now, we have two phase 2 studies ongoing. Phase 2 studies are the initial patient studies, and I'll tell you more about those in a moment. We also have a very strong patent portfolio, and we have significant R&D experience within our team. Both myself and two other key individuals on our development team have had the opportunity and the fortune to deliver 3 drugs through the FDA for approval. Next slide. So this is a snapshot of our portfolio. The first 2 lines are our clinical trials in MSA. And as I mentioned, those are phase 2 studies in patients. The first study is the definitive study I'll call that 201. We completed enrollment in that study in November, and we'll be completing that study in November of this year. Next is the 202 study. That's in a somewhat more advanced patient population than the first study I told you about. And that's an open label study, and that has the benefit of giving us an early indication of efficacy in the middle part of this year. We reported in December some exciting new data in an animal model of Parkinson's disease, a primate model. That's a collaboration with the Michael J. Fox Foundation. I will be telling you more about that. And then this BioMuse study, towards the bottom of the slide, has been an incredibly valuable study that we've been conducting in cooperation with Vanderbilt University here in the United States. Next slide. So wherever there is a big unmet medical need, as there is in treating MSA, there also happens to be a substantial commercial opportunity, and that's the case with 434. We've done marketing research with US neurologists, both generalists and specialists, and based on relatively modest assumptions in terms of a treatment benefit, orphan drug pricing, and expected uptake, we estimate peak sales to be over $1 billion. That's just in the US, and that's just for MSA. And there's a lot more behind this, but the clinicians clearly had a strong intent to prescribe and really appreciated the fact that we had an oral drug that was addressing the underlying pathology of this disease. Next slide. So I'll be talking about Parkinsonian disorders. which are named for the fact that they present with Parkinson's-like symptoms. I think you've all seen someone with Parkinson's disease who has slow movement, maybe a shuffling gait or tremor. And it gets its name Parkinsonism because Parkinson's disease is the most common cause. Now, on the next slide, you'll see that there are other disorders like our target disease, MSA, that are also considered Parkinsonian disorders. And what's important here is that both Parkinson's disease and MSA have similar underlying pathology. Next slide. So this is a study of the brains of patients who have either Parkinson's disease on the left or MSA on the right. And these researchers basically took their brains and studied the areas of pathology and characterized the amount of iron in those areas. And the patients are in blue, and in green are healthy controls of the same age. And we see both in Parkinson's disease on the left and in MSA, or multiple system atrophy, that there are multiple brain regions that have increased iron. And this is really what we're targeting with our therapy. We're really trying to reduce this iron and reduce the ongoing damage in the brain. Next slide. Now, if you fast forward about 25 years from when that study was originally done, our collaborators at Vanderbilt and others have developed and are really perfecting advanced MRI methods to measure the brain iron in these regions of pathology. So if you see at the tip of those orange arrows from the brain study, if you then look at the lower left-hand corner, the orange arrow there is pointing to the same increase in iron in the brains of a living patient. And that's important for two reasons. Number one, it helps us identify the patients with a fair amount of precision. And number two, it helps us quantify the change in that iron with our therapy. Next slide. So iron itself is a strong contributor to pathology, and it does it in two different ways. Number one, you see on the left is a cartoon of a really important protein called alpha-synuclein, and that's important for neurons to communicate with one another. So with excess iron in the brain, it actually causes that protein to clump, and therefore it can't function. That's one problem. The clumps then the body tries to clear them, and that leads to an inflammatory response. And the excess iron also drives oxidative stress, and you see this all on the right. So what we're trying to do is to reduce this iron imbalance, lower the iron, and eliminate the source of this ongoing damage. Next slide. So what our general approach is summarized on this slide, where we redistribute excess iron in the brain. We reduce the alpha-synuclein clumping and the oxidative stress that damages intracellular structures, and thereby rescue neurons. And the overall goal is to potentially modify the course of disease. Next slide. So here's the cartoon of or a model of 434 on the right. It is a small molecule, which means you can orally administer it, and it is absorbed and delivered to the site of action. That's really important because we've shown that in multiple animal models as well as in man in our phase 1 studies. As mentioned, this has potential to treat various Parkinsonian disorders, and we have been discussing our regulatory approach with the FDA and European agencies who have endorsed our approach. Next slide. I want to now tell you just a little bit about one animal study that we recently conducted. And this is a primate study or monkey study. And this is a really important study because primates are closer to humans than other animal models typically used. Now, what this study showed was that 434 improved the motor skills and the general behavior in monkeys that had experimentally induced Parkinson's disease. And that's important because these animals, the monkeys, really behave a lot closer to human behavior than other laboratory animals like rats or mice. Now, the other important point here is that those animals that had the greatest improvement also had lower brain iron in the area of pathology. And that's really important because that increases our overall level of confidence in the ongoing phase 2 trials where we're targeting iron in order to reduce pathology. Next slide. So I don't have time to go through all these studies. That monkey study I just told you about is on the bottom of the slide in blue. But I think what you see is that all the studies that we've conducted over several years show that we can lower brain iron, reduce that alpha-synuclein protein clumping so it can function normal, and improve the number of neurons or the connectivity and improve motor performance. And this is really important to give us the reason to believe to go into the clinic with our drug. Next slide. So I'd now like to pivot and tell you a little bit about our clinical development program in MSA. Next slide. Now, this is a rather busy slide. So if you focus on the first column, this is the natural history study that we started in 2020. And the overall goal of the study was really to design and de-risk the phase 2 study. And that has continued to reap incredible benefits in helping us design the right protocol to identify the right patients and also to select the right biomarkers to put in those studies. Now, the 2 studies in the orange box, those are the phase 2 studies that I told you about at the beginning. Those are in MSA patients, and there's treatment here. So patients are either receiving 12 months of, in the first study, the 201, in the middle column, they're going to get 2 doses of 434 placebo. In the study on the right, they're just going to get 1 dose level of 434. Now, what's quite important is in the green box, and that's based on all of our preclinical studies that we have identified the main endpoint to declare success is change in brain iron as measured by those specialized MRI techniques. So the definitive trial is in the middle column. The trial on the right is the open label trial, but that can give us an early indication of efficacy before the double-blind trial reads out. Next slide. So I just wanted to review key milestones going forward. I think I mentioned at the top that we completed enrollment in the 201 study in November. And that study, if you look on the far right in November of 24, that's when the study will complete. And we expect to deliver data in the next 2 to 3 months after that. And I also told you about the monkey data in Parkinson's disease that read out in December. So in addition to the double-blind trial reading out late in 24 or early 25, the open label study in MSA patients, we expect to have preliminary data from 6 months of treatment in the middle of in the middle of this year. And then the next slide, I believe, is my last slide to just summarize that we are really poised for progress in 2024. We achieved all our clinical and corporate milestones in 2023. Our first indication is an orphan disease, MSA, with no approved treatment that has substantial commercial opportunity. The new primate data really validates our clinical strategy. And I'm very proud of our development team with the 3 FDA approvals that we have under our belt. And with that, I will stop and thank you for your attention. Look forward to your questions.
Thanks, David. Bang on time. Really appreciate it. Had a lot to get through there and really fascinating. We know how tough Parkinson's disease is, and it's great to see the work that you're doing. Got a few questions here for you. You recently reported some data that indicates the efficacy of 434 in a model of Parkinson's disease. Can you just elaborate a bit more on these results and the implications?
Yeah, so that was the primate study that I was explaining a little while ago. I didn't have time to go through the data themselves, but I guess what I'll elaborate on a little bit is why that study is so important. Number one, primates are a higher order animal compared to rodents, where many animal studies are done. They have behaviors that are much similar to humans, so we can assess their ability to climb, their balance, their appearance, their ability to interact with veterinarians and things like that. So those are things that are much more relevant to how humans are assessed in the clinic. And also we can assess their motor behavior in a more systematic manner than we can in smaller animals. So that study was really so important because it's closer to humans. And it really showed that in those animals that improved, we lowered iron with the drug, which is really what we were hoping to do.
Gotcha. And how important is the BioMuse natural history study, and what additional value can it add?
Yeah, so that study is one I'm very proud of, and I have to really tip my hat to our collaborators at Vanderbilt, who have done such excellent work. MSA is a clinical diagnosis. So you really have to base it on clinical symptoms and on physical exam, but it's a notoriously difficult disease to diagnose. So it's very important that you identify the right patients to come into your clinical trial. And what we really learned from the BioMuse study is that many patients who look like MSA actually have Parkinson's disease. And what we learned from the MSA study, or from the BioMuse study, was how to really fine-tune our protocol so we wouldn't enroll those Parkinson's patients into the study. You can imagine if you, because Parkinson's disease progresses much more slowly than MSA. MSA actually progresses about 3 times as rapidly. So if you get too many patients who have Parkinson's disease in your trial, and they should happen to be enrolled into the placebo group, they'll look like they're doing better because they're not declining, when in fact, it's not because of drug or placebo or whatever, it's just because they have a different disease. So we identified really how to enroll those patients with precision. And it also really helped us identify how to measure the iron in the brain with a great amount of precision. And that's really why the BioMuse study is so important.
And what other applications might 434 be considered for?
Yeah, well, 434 is a very interesting drug. I think many neurodegenerative diseases have increased iron. We know about Parkinson's disease. We know about MSA. There's one other orphan disease on my slide called PSP that has no approved treatment. Many investigators believe that increased iron is associated with the underlying pathology of Alzheimer's disease. And we've made disclosures about patents that are targeting new molecules for diseases like Alzheimer's and Parkinson's. And then there are other rare diseases. Friedreich's ataxia is another one where there's increased iron in areas of pathology. So this is a very ripe area for developing new therapeutics. And there are incredible, as I'm sure all the listeners know, all those diseases have very substantial pathology, and there's a real great need to address them.
And just to close up, David, with regards to the investors online today, what sort of news flow can they expect over the coming months and in 2024?
Yeah, so I think the most exciting data that's coming is in anywhere from the March timeframe through the June timeframe, depending on when the enrollment settles out and when we analyze the data. And that's actually probably on the right side of the slide, the upcoming catalyst. We're getting preliminary data from that open label study on the iron content in the brains of those patients. And that's really, we expect that by the middle of the year. We've got more data presentations coming up at congresses later this year. We'll be announcing that more specifically later. We're continuing to pursue science with our collaborators, and we expect collaborations and future collaborations, research, publications, et cetera. So there's a lot to look forward to in the coming year.
We'll watch the story closely. Thanks for your time today, David, and have a great weekend, and appreciate you calling in from San Francisco.
Great. Thank you, Paul. Thanks to all the listeners.